I would like to thank, of course, our sponsors, especially the BBLA, the Bohemian Benevolent and Literary Association, and also the Czech Ministry of Foreign Affairs, which has given grants especially to support our efforts to record and put online an archive of our events. And we would like to thank all of the individual donors, many of whom are here among us. We encourage you to follow us on whatever platforms you're using, and especially check out on YouTube, where you will find the archive of past programs. If you enjoyed this evening's program, you might find other things in there you would find interesting. And I would like to invite you to our next event coming up on Thursday, May 1st at 7 p.m. in this very room. Our own board member, Vít Hořejš, is going to be leading a recollection of a remarkable event in the history of Prague when Allen Ginsberg was elected the King of May for a student festival that they had on May 1st at that time, and then got himself in trouble. And our board member, Vít Hořejš, was a witness to that event as a teenager, and he and some other people who know things about that event are going to help us reconstruct that. And we encourage you also to come for this one, part of the same series, the Pressburger Klezmer Band from Bratislava, and Frank London with the Old Bohemians, aka Polka Loka. Our speaker today, Hynek Wichterle, is a professor who holds a joint appointment in the departments of pathology and cell biology and neuroscience in neurology at Columbia University Medical Center. He received his master's degree from Charles University in Prague and his PhD from the Rockefeller University. He trained after taking the PhD at Columbia University, where he became an assistant professor in 2004, an associate professor in 2012. He serves as a co-director of the Columbia Stem Cell Initiative and as a vice chief of the Division of Regenerative Medicine in the Department of Rehabilitation and Regenerative Medicine. I realize that I met Hynek 30 years ago, which, if you can imagine, we were both even sexier than we are now. And it's just amazing that he is where he is, and so I'm so delighted to introduce him, and I've seen him do this shtick before, don't be intimidated if you're not a biologist. He's going to do it in a way that I think we'll all be able to understand. Please welcome Hynek Wichterle.
So thank you very much, Chris, for this generous introduction, and thank you to SVU for bringing me here and giving me the opportunity to torture you for some time about very basic topics that are dealing with developmental biology and stem cells and how we are trying to use stem cells for the benefit of humankind. Chris already introduced me, so the only thing that I want to mention is, for conflict of interest, is that I'm a co-founder of a new biotech startup, which is called Progenex, and some of the work that I'll be presenting is now being developed in the startup for clinical trials. So I'll have to start with a little bit hard slide, and that slide shows you one of the biggest upcoming challenges for our generation and the younger generation, which is the realization that actually as the population ages, there is increasing burden of diseases, late onset degenerative diseases that are burdening not only people who are affected by them, but also financially the medical system. And so you can nicely see it on just this pie chart that summarizes the costs of Alzheimer, of treatment or taking care of Alzheimer's patients annual. And Medicare only is contributing $157 billion annually to care of Alzheimer's patients. It's currently 21% of the Medicare budget, but because the population is aging and it's estimated that by 2050 the number of people over 70 years old will double, it is estimated that the cost will rise to $360 billion. And that will consume pretty much half of the entire Medicare budget. Now, recently there was a calculation that suggested that if we could simply just delay the onset of Alzheimer's disease by as little as 5 years, that would translate into very significant saving for Medicare. It would save around $140 billion per year. And this would make a big difference. So it's really frustrating that we are still in a situation after so many years where we do not really have any effective treatment that would not even stop the disease, but even just delay it by several years. And this is just Alzheimer's disease. We have to realize that besides Alzheimer's, there are many other diseases, such as Parkinson's disease, affecting the dopaminergic neurons, Huntington's disease, frontotemporal dementia, and amyotrophic lateral sclerosis, which is the disease that I'll be focusing on during my presentation here. So while Alzheimer's disease is really affecting our cognitive functions, amyotrophic lateral sclerosis, on the other hand, is affecting mostly our motor functions. And so what are motor functions and what are motor neurons that are degenerating in this disease? It turns out that motor neurons are actually, so the motor pathway starts in the motor cortex, where people initiate the planned movements, and then the cells found in the motor cortex, they have long projection that goes all the way to spinal cord, and in spinal cord these cells make a synapse onto spinal motor neurons that then send axons outside of the central nervous system. They innervate our muscles or organs, and they control all our movements and also visceral functions. So the fine motor skills that are required for Iva Bytová to play her violin, and they are controlled by these motor neurons, as well as the sequences of movements that are necessary for Jaromír Jágr to succeed in his hockeys. So it's really important, this system, and actually the patients that in whom this system starts to degenerate, it's a really tragic situation because they are cognitively often completely fine, and what happens is that they just watch themselves to lose ability to control their body. Eventually they lose their ability to breathe, and they die. The disease was initially described by Jean-Martin Charcot in Paris in 1869 as a progressive weakness, loss of motor control, and the average survival after diagnosis is now around 4 years. The disease is, although it's pretty rare, it's not as rare as we think. So actually, a lifetime risk of contracting or succumbing to ALS is 1 in 300 people. And unfortunately, for 120 years, we really did not have any insight into what is causing this disease until actually Bob Brown, at the time at Harvard University, discovered in a family that had a hereditary form of ALS, a mutation in a gene. He identified a gene called SOD1, and he identified there are actually a number of mutations in different patients, and realized eventually that these mutations cause actually misfolding of the protein. And the misfolding of proteins is something important because actually pretty much all degenerative diseases, from Alzheimer's, Parkinson's disease, all the way to ALS, are often associated with accumulation of misfolded protein in the cells that are dying. So just to explain what I mean by misfolding protein is proteins are basically sequences of amino acids that can be hundreds of amino acids long, and this string of amino acids has to fold often into some complex globular or folded structures. And if the string falls in a wrong way, the protein will not be functional, and often the cell recognizes it as a bad protein and has to get rid of it. And so somehow these mutations often cause protein misfolding, and these accumulated proteins in the cells lead to the degenerative disease eventually. Unfortunately, despite actually knowing and understanding some of these etiologies of ALS, we have now more than 20 different genes identified that patients with inherited forms of ALS in which we can find mutations, and often these mutations, as well as in SOD1, cause protein misfolding. There were in excess of 60 clinical trials now for different treatments for ALS, and out of that there is currently only 3 approved therapies, and each of these therapies only prolong survival by several months, so there is nothing really effective. So we can sort of summarize this timeline as 150 years of failures to try to understand and treat the disease. So during my presentation, I will touch on two questions that in my mind are really at the heart of the problem of many neurodegenerative diseases. So the first question is that, I mentioned that a subset of ALS is caused by mutations that are inherited in families. And remarkably, these mutations or the genes that are mutated are present in most cells in our body, yet only motor neurons are sensitive and they degenerate in the disease, and we don't really understand why other types of cells are resistant to these mutations. So that's one very important question. And then the second question is that these mutations are also present in the cells throughout the lifetime, yet these diseases develop only later in life. So you can be born with a mutation that will cause ALS. You will have no idea that your motor system is impaired in any way. You can actually be excellent athlete like Lou Gehrig, and then eventually your system starts to degenerate and motor neurons die for some reason. So we don't really understand what is contributing to this late onset. And so in order to start understanding what underlies the motor neuron degeneration, I believe that we really need to understand what makes motor neurons different from other cells in our body. And, but that is and has been always problematic, because motor neurons are found inside the vertebra, so they are not accessible. You cannot watch them. You cannot make biopsies and remove them, because that would be detrimental. And so the only way how people studied this disease for many years is by looking at post-mortem samples in deceased ALS patients. But recently we started realizing that there might be other ways how to access this cell type, which is not very accessible. And that's by learning from the normal development that happens in the embryo. So we know that, that all cells in our body originate from a single fertilized egg. This egg has to divide, and somehow during the embryonic development, some cells have to be instructed to become motor neurons. And so just to illustrate you how beautiful this process is, I'll show you this video of developing a frog fertilized egg. You see that the cell starts to cleave. The cells are dividing pretty symmetrically, so they all divide at the same time until they form this ball of filled with many different cells. But then something happens. At this point, these cells still look very similar to each other. But then something happens, and this ball of cells starts to undergo gastrulation. This is a process when different tissues become separated and they acquire distinct identities. And you can actually see that at this point, there is a part of the tissue that folded and formed the initial neural tube that will give rise to the entire central nervous system, with the head developing here and then the spinal cord developing in this region. And so how these cells become instructed to become different parts of the nervous system, or nervous system versus skin cells or blood cells, this has been something that scientists studied for now many decades. And the idea is that if we understand what instructs, what educates cells, what type of cell type they should differentiate during development, we might be able to recapitulate the process and make our own motor neurons. So in order to make our own motor neurons, there are two conditions that we need to fulfill. First, we have to have access to naive cells that are pluripotent, meaning that they can give rise to any cell type in our body. So we have to be able to grow these cells and use them then for the differentiation. And then the second problem is that as these cells divide, we have to be able to identify or know what are the signals that instruct the development and cell identity of these cells as they undergo development. So the first problem has been actually solved now quite a few years ago, where researchers such as Gail Martin and Martin Evans identify conditions under which They can take actually relatively young developing embryo. So they started with mouse embryos 4 to 5 days old. That looks like this. This is basically a little hollow ball with a set of cells inside that will give rise eventually to the whole embryo. And when you take these cells, they found conditions under which you can grow them in a petri dish. These cells divide and they can grow, basically you can grow unlimited amounts of these cells. Now, we can even get by without relying on these early blastocysts. Actually, Jamie Thompson found ways how to take blastocysts that are discarded from IVF clinics, from couples that already went through pregnancy and they do not need any more pregnancies. And they took these discarded blastocysts and actually showed that they can derive also human stem cells like this from early blastocysts. But there's always ethical issues with using even discarded blastocysts. And so most recently, actually researchers such as Shinya Yamanaka found ways how to take other cells from adult organisms. So we can take a piece of skin or blood cells and then actually reprogram these cells into cells that have the same properties as embryonic stem cells. And so these embryonic stem cells are amazing because, as I said, you can produce unlimited supply of them. They can grow for years in a cell culture in petri dishes. They are pluripotent, which means that they can give rise to every single cell type in our organism. So they can give rise to every type of neuron, skin, heart, muscle, anything. And finally, because we can grow these cells in cultures, we can also engineer them quite easily. We can add genes into them or remove genes and study what these manipulations, what effect they have on the function of these stem cells. So now we have cells that from which we could produce motor neurons, but do we know how to make motor neurons? And that's the second problem that people solved, or developmental biologists solved over several decades. So this is a top view of actually developing chick embryo. This would be the brain inside the head. And then here you see this long tube, that's the developing spinal cord. And then if you cut cross-sections through the spinal cord, so this is relatively early, so it looks like a tube that has an opening inside. You can basically see all the different cells that will give rise to different types of neurons in the spinal cord. And importantly, researchers identified several signals that are important in specifying cells to become spinal cord. Actually, one signal is a derivative of vitamin A called retinoic acid that is secreted from this region called paraxial mesoderm and instructs the cells in the vicinity that they should become a spinal cord tissue. And then in a sort of similar way, like in a Cartesian system where you have to specify rostrocaudal and dorsal ventral axes, you also have to specify different cell types along the dorsal ventral axis of the neural tube. Motor neurons are actually born very ventrally in the cells, which means like on the belly side of the spinal cord. And it has been found that there is a molecule or protein which is secreted from the ventral floor plate and notochord, which has been named sonic hedgehog. And the cells that are exposed to high concentrations of this sonic hedgehog molecule will adopt motor neuron identity. And so what I wanted to know is whether we actually know enough about the signals that control motor neuron specification to allow me to actually recapitulate aspects of normal embryonic development in a petri dish. And so for that, we took advantage of engineered mouse that expresses fluorescent protein, green fluorescent protein in all spinal motor neurons. So this is the head of the developing, this is embryo of the mouse. So you see actually it's pretty translucent, and you can see the green fluorescent light here are all the cell bodies of motor neurons. And then you can see also the axons which are growing to innervate different muscles. So we took these mouse embryos, we derived embryonic stem cells from them, and then I exposed them to different combinations of the signaling molecules that I already introduced to you, and found the precise recipe that allows me to convert the embryonic stem cells that grow in these clusters floating inside the medium in the petri dish to induce them very efficiently to give rise to motor neurons. And not only that these cells at the end turned on the green fluorescent marker suggesting that they became motor neurons, but we could even take them and test their functionality. And for that, what I did, I actually decided to transplant them back into the developing spinal cord and see whether they would be able to integrate into the nervous system and perhaps innervate muscles. But that would be very difficult to do, to take mouse cells and put them back into mouse because the manipulations would have to happen inside the uterus. So instead, I decided to take the risk and see whether these cells can be transplanted into developing chick embryos. And so here, I made openings into fertilized chicken eggs. And then I made actually little lesion. I removed part of the chicken neural tube and implanted back the mouse-derived motor neurons. And remarkably, these cells not only survived inside this different host, but they also started growing axons to periphery. And when I followed these axons, the projections, all the way to the sites where they contact muscles, I could see these specializations called neuromuscular junctions that actually send the signals from the motor neuron to muscle and instruct the muscle to contract, suggesting that these cells are actually fully functional. So this was great. We had now plenty of mouse motor neurons, but eventually to understand human disease, we would like to have also access to human motor neurons. And that turned out to be actually really challenging. It took us, us and other investigators, including my former postdoc, Stefan Nedelek, 15 years, and he moved at the time to his own lab in Paris, where he actually investigated different sets of signaling molecules that we might be missing until he discovered the one additional factor that we need to add to human cells in order for them to differentiate effectively into motor neurons. But now we have as efficient, or maybe even more efficient system to make motor neurons from human embryonic stem cells or pluripotent stem cells. So this was really satisfying because I was always inspired by a sentence that was left on Richard Feynman's blackboard at the time when he died that says, "What I cannot create, I do not understand." And so my feeling was that if we really understand how motor neuron identity is specified during normal embryonic development, we should be able to create motor neurons in a petri dish. And by showing that we can make motor neurons, I think that there was a very strong confirmation that we understand the process in great detail. But then, obviously, it was much more interesting for me to try to understand, answer the two questions that I posed in the beginning of my presentation. Why are only motor neurons dying in ALS? And so I take you very briefly through several years of work in my lab, where we started asking whether motor neurons show any increased sensitivity to some kind of stressors that we can expose them to. Asking basically whether we can identify what makes motor neurons more vulnerable compared to other neurons. And so for that we actually used, so this is, you're now looking into a petri dish with many different cells growing in it. Each of these blue dots is a cell nucleus, and then the green cells are motor neurons. You see the cell bodies, and then the processes, the axons and dendrites are growing from these cells. But in this experiment, we mixed in also spinal interneurons, which is another type of neurons that helps to communicate between, in neural circuits from one circuit to another. And remarkably, when we expose these cultures to a set of toxins, actually toxins that specifically induce protein misfolding, you start seeing a situation where the motor neurons, the green cells, are basically disappearing from the cultures, but the red cells survive. And so this was really interesting. So in this case, we are exposing it to a mycotoxin is called cyclopyrazonic acid. But to us, that it really highlighted that motor neurons are much more sensitive to induction of protein misfolding compared to other types of neurons. And that could explain why some of the mutations that cause protein misfolding in genes that are expressed or present in all cells in your body would be preferentially toxic to spinal motor neurons. And so having this system where we could basically kill off our motor neurons, we can then turn it around and start asking, can we use this to discover new molecules that would make motor neurons behave more like these interneurons, that they would be resistant to these levels of toxic exposure. And so for that, we joined forces with a chemist at Columbia University, Brent Stockwell, who synthesized around 120 new compounds for us. And Emily Lowry in my lab screened all of these compounds plus 2,000 of other existing compounds to try to identify families of small molecules that would be protective to motor neurons. And remarkably, together with Brent and Emily, we found one compound that Brent actually synthesized that is very potent in protecting motor neurons. And you can see it again. This is looking now at motor neurons, are these black dots with their processes growing out. So this would be control culture looking in the petri dish. Now you expose the cells to the toxin, and 2 days later you see that most of the cells died and the processes shriveled or they fell apart. But now if you expose them to the toxin and add this small molecule inhibitor, you see that you can very effectively protect pretty much all motor neurons in the dish. And so we are now taking this new molecule that is not approved for use in patients, and we had to do a lot of toxicological studies and got a few years ago approval from FDA to start clinical trials. And actually initially the FDA allowed us to only start at relatively low concentrations because they were worried of potential toxic effects of this drug in patients. But that just 2 weeks ago they lifted the ceiling for us, so we can now go to concentrations that we believe would be efficacious. And the plan is to start dosing first ALS patients probably sometime this summer with increased concentrations of this drug. Of course, this is just a first step, and from the history I told you that there is more than 60 trials that failed, so there is no guarantee that this will be successful. But what we are hoping is that maybe this drug could become a part of multi-drug sort of cocktail that will be eventually used to promote survival of motor neurons and perhaps other types of neurons in patients. So this takes me from, I would say, the more sort of grounded scientific part of my talk to the part where I get into a little bit more esoteric territory. And that's our ways how we are now trying to think about other possible avenues how to deal and treat diseases of neurodegeneration. So this is the part of where we get into the alchemy of the research. So what is alchemy? Alchemy is the search for philosopher's stone, and philosopher's stone has been described as a material that has ability to actually transmutate elements, i.e. turn one element into another element, and also that it can serve as an elixir of youth. And so what is transmutation? What does it mean? So transmutation is described as a process of changing the structure of an atom's nucleus, which results in a change in the identity of the atom. So, it sounds very opaque, but it's not something that does not happen. Actually, alchemists, although we sort of think them as little hacks, they were serious about this. And in reality, actually, transmutation is happening all the time inside the sun, where hydrogen is fusing into helium. So you are basically transmutating hydrogen atoms into helium atoms, or during fission, when plutonium is basically breaking into uranium and other elements. But the other thing that is quite interesting in this description is that the effects are on the nucleus of the atom. And actually, even cells have their nuclei. So this is, again, motor neuron. This part is what is called the nucleus. And nucleus is the site in the cell that actually contains all the genetic information. The 3 billion of letters of our DNA are packed into this small structure, the 6 feet of the double helix strand. In these 3 billion letters, they encode 20,000 different genes. And what are genes are stretches of DNA that basically define the sequence of proteins, the amino acid sequences that I described before. And so what differentiates one cell type, motor neuron, from your blood cells? Actually, it's not that each cell would have different set of genes, but in each cell, different set of genes is turned on and off. So basically, each cell selects only subset of these genes to turn them on, and that defines the function and identity of individual cells. So if you look at a piece of a chromosome or DNA strand, so this would be maybe something like 1 million of these DNA letters, you see that within this region of the DNA there are several different genes. And as I said, in each cell, a different group of these genes will be turned on. Now, how do cells actually control the genes, which gene they will turn on or off? That's, to me, like the most beautiful problem in biology, because actually cells use products of genes to control their own genome. And these genes are called transcription factors. They are proteins that actually can recognize sequences in the DNA and can actually find these sequences among the 3 billion nucleotides or DNA base pairs in the genome and sit at very specific locations. And so you can also notice that most of the DNA does not contain genes. It's this empty space, and that's exactly where these transcription factors bind. We have around 1,639 transcription factors as the latest count. And basically, which of these factors are turned on in a cell will define which regions of the DNA they will occupy, and then also which genes they will regulate. This sounds sort of very simple and mechanistic, and suggests that we should be able to predict how transcription factors control DNA, but in reality, actually, it's not the case. So you can think of these transcription factors as these little philosopher's stones, because they can, when you put them in the nucleus, they will change the organization of the nucleus, the structure of the nucleus, and they will change the identity of cells. But the effect of these transcription factors is still really unpredictable for us. We cannot computationally say that adding this transcription factor will convert skin cell into a motor neuron, for example. And so a lot of these studies relies on empirical investigation, where we add one transcription factor to a cell and ask how cell responds, or take the factor away and ask how cells respond. Similar like the alchemists when they were mixing different molecules and heating them and seeing how the metals respond. So actually, we were quite interested in understanding what transcription factors would turn on the genes that define motor neuron identity. And that's work that was done by a former postdoc in my lab, Esteban Mazzoni, who's now at NYU. And he basically, as an alchemist, started testing different transcription factors that he added to embryonic stem cells, these pluripotent cells, and asked whether he can find combination of factors that would reprogram or transmutate these embryonic stem cells into motor neurons without relying on the normal developmental pathway that we relied on before. And he actually focused his attention on two factors. So this is, again, a cross-section of the developing spinal cord. This is where motor neurons are born. And there are two factors that are very highly present or turned on, specifically in the location where motor neurons are being born. And these transcription factors are called ILED1 and LHX3. And remarkably, when Esteban built and engineered an embryonic stem cell line in a way that he can turn on these two transcription factors in the cells, at his will, it turns out that when you activate these two transcription factors, within 48 hours, so very rapidly, these cells basically convert from rapidly dividing small cells into motor neurons. And it happens very efficiently and also very rapidly, which was quite remarkable, because that shows us that we actually identified the key factors that can turn on the right set of genes in order for these cells to become motor neurons. So this was great, but, and yeah?
I'm so sorry.
No problem.
You need both transcription factors?
Yes, you need both factors.
One or the other doesn't do
No, one or the...
Can be added sequentially?
So we have not tried to add them sequentially, but we know now, so we also mapped how they bind to the DNA, and we know that they actually bind together to the same sites in the DNA. So we think that they form actually what we call heterodimer. They bind to each other, and then they sort of identify the right locations in the genome to activate the genes. this was really exciting because it showed us that eventually we might be able to find combinations of transcription factors to produce almost any cell type in our body that could be of clinical relevance. But eventually we actually turned out to another question, and that question was related to the fact that, or to the question of why motor neurons are susceptible to disease only when they reach certain age. And so that brings me to the second part of the alchemy, which is the elixir of use. So I told you that transmutation happens relatively, is something that happens in biology, in nature, and it's relatively easy to justify. But elixir of use was always something that people joked about, that alchemy never actually showed that something like that could exist. But we think that with these transcription factors, we might be eventually able to actually change not only the type of cells that you produce, but also age of these cells. And so this is work that started in my lab by talented postdoc Tulsi Patel, who just last summer moved to her own, to start her own lab at the Rutgers University. And what she asked is how motor neurons change from the time they are born early in the embryo all the way in aged organisms. And she asked, are these motor neurons very quickly, do they turn on the right set of genes, and as soon as they turn on the right set of genes, they maintain the same set of genes through the lifetime, or do these cells change as the organism matures? And so she isolated motor neurons from all these different stages from mice, and then asked what genes are turned on or off in these cells. And so now I'm representing her data in sort of this more abstract way, where you basically can ask which of the 20,000 genes are expressed, and then just show distance how different the expression is from one state to another. And so this would be how the cells in the embryonic day 10 mouse looks. This is 3 days later, embryonic day 13. This is now postnatal day 4 motor neurons, postnatal day 13 motor neurons, and then postnatal day 21, 56, and 2 years. And so what you can appreciate here is that there is a remarkably dynamic change in gene expression all the way from embryonic day to postnatal day 21. And at this point, at postnatal day 21, the motor neurons become very stable, and they do not really change during the rest of the adult life. And so that suggested to us that there has to be some kind of program, gene expression program, controlled by some juvenile transcription factors that activates this youthful program in motor neurons, and then there has to be some adult program which is activated by postnatal day 21 in these animals that establishes the adult motor neuron gene expression program. And actually Tulsi was able to use other tools that I probably should not really get into here, but she was able to identify what families of transcription factors are controlling the adult state and the embryonic state. And remarkably, while she found that the adult state is controlled often by neuronal activity, which means firing action potentials, as well as exposure to hormones, different hormones in a maturing organism, the juvenile program was controlled by actually our well-known factors, ILAT1 and LHX3, that define the identity of motor neurons early on. These transcription factors, so I already showed you that these two transcription factors are sufficient to convert embryonic stem cells into motor neurons, but remarkably, very soon after motor neurons become born in the embryo, these factors are downregulated. So the cells actually stops producing these transcription factors, and then the cell goes through this maturation and aging state when the motor neurons become susceptible to the mutations that cause ALS. And so we actually asked a relatively straightforward question: what would happen if we re-express the factors in the adult motor neurons? Would we be able to actually change the state of these cells and reactivate some of the more immature properties in these cells that would make them more resistant to the mutations and the accumulation of misfolded proteins in these cells? But this is not, it's very easy to write it on a blackboard, but it's not actually that easy to do it in biology. So actually what you have to do is you have to start by isolating the ILAT and LHX3 genes. So you have to find the stretches of the DNA. you have to usually put them in bacteria. And then we had to actually change the regulatory sequences that control in which cells the genes are turned on. Because these genes are turned on in adult motor neurons, so we had to find other regulatory sequences that would allow these genes to be turned on in adult motor neurons. And then how to get them into adult motor neurons, we had to package them actually into viral particles. We used virus, which is called RNA-associated virus, that actually if you inject into animals, it will infect very effectively most of the neurons. And then after we infect these mice with the viruses, we have to check that the genes are turned on in motor neurons, and very selectively in motor neurons, because we did not want to reprogram other neurons into motor neurons. That could be quite bad. And so here is the viruses that we produced, and we injected them actually in newborn mice. And then here you can see again, this is a cross-section of spinal cord. And now you see this is adult spinal cord. And normally you would not see any of these red and green dots. So each of these dots is actually nucleus, and the transcription factors go into the nucleus where they control gene expression. The blue delineates the area where motor neurons are found. So this is the ventral part of the spinal cord. And you can see that the transcription factors are highly expressed in the nuclei of these motor neurons after we infected these animals, suggesting that the tools he developed worked really well. And so now with this in our hand, we wanted to know what happens if we now infect motor neurons in a mouse that carries the mutations that cause motor neuron degeneration. And so we used a mouse model of ALS that actually expresses the SOD1 protein that I said is mutated, was found in one of the first patients. So this mouse expresses the mutant SOD1 protein. And normally in this mouse, you start seeing aggregation of misfolded proteins already on postnatal day 45. And then you start seeing actually loss of motor neurons and behavioral deficits. So the mouse stops moving by postnatal day 90. And at 150 days, the mouse dies, while normal mice live for 2 years. And so in the first set of experiments, we injected the virus at postnatal day 1 and then asked whether we can see any changes in these misfolded proteins. And so this is now looking very high magnification. So this is a cell body of one motor neuron. This is the nucleus. This would be another motor neuron, another motor neurons. Now this is a healthy motor neurons, and the green staining is for actually structures that degrade misfolded proteins. And in healthy cells, you see these little small dots that are each basically degrading misfolded proteins. But in the case of ALS, you start seeing these pathogenic large aggregates that accumulate inside the cytoplasm of these motor neurons. So when we then added the virus, so we added first virus that expresses control genes. In this case, it was red fluorescent protein. And you see that still these motor neurons that express the red fluorescent protein have these large aggregates of misfolded proteins in them. But remarkably, when we now express ILAT1 and LHX3 shown in red, so again, these factors, because they're transcription factors, they go into the nucleus where they bind to DNA, and they change the gene expression program in these cells in such a way that these green aggregates look much more like the aggregates that you found in a healthy organism. So that to us really suggested that we were able to make these motor neurons somehow capable to handle the mutant proteins as well as young motor neurons would. And so this gives us a hope that one day we might be able to find ways how to actually rejuvenate adult motor neurons and maybe help them to fight the mutations that cause degenerative diseases, not only in this case that would drive from ALS, but in other neurodegenerative diseases. So this brings me to the end of my presentation. And I hope that you will be able to take away a few messages that if you think about why motor neurons are susceptible to the disease, not other types of neurons, it might be because motor neurons are more sensitive to accumulation of misfolded proteins, and that we identified actually a pathway that is called MAP kinase pathway that these cells activate that leads to their degeneration. And the drug that we developed together with Brent Stockwell inhibits this cascade, this pathway, this signaling pathway inside the cell, and prevents or stops motor neuron degeneration. And then in the second part, I hope that I was able to show you that motor neurons undergo this very prolonged maturation where their gene expression program changes. In mouse, it takes 1 month of their life, which, in human, it could be easily equivalent to, until you become a teenager. And then during this process, the motor neurons become less capable to handle misfolded proteins, and you start seeing an accumulation of misfolded proteins as soon as the motor neurons reach the mature age. And that we have now ways how to actually change the age status of these motor neurons, how to reprogram them into more immature state, or state in which they are more resistant and they can clear the misfolded proteins. I think I acknowledge most of the people that contributed to this work, but Emily Lowry, John Costa, Esteban Mazzoni, Tulsi Patel, Stefan Nedelek, and Sebastian Thams were the main people from my lab that generated all of this wonderful data. And I would be happy to answer any of your questions if you have. Thank you.
Thank you so much, Hynek, for taking us into the alchemical world. I think I got most of it, but some questions from our audience.
Fascinating, from what I understood. First, 2 questions. The first one is, you mentioned 150 years of failed experiments. So what keeps you optimistic, motivated, waking up in the morning? And the second question is, with all this fascinating work you've done, have you found any of this helpful for other neurological diseases?
Mm-hmm. Yeah, these are great questions. So what makes me optimistic is, it might not be that I'm optimistic about the work that is coming from my lab, but I'm optimistic about the field in general, because actually we are living through almost a revolutionary time in biological research or biomedical research, because the abilities for us to sequence genes, to understand, as I said, where transcription factors bind. 10 years ago we had pretty much zero idea where transcription factors bind in the DNA, and now we have the tools to map this, to predict, and slowly get to a point where we will be able to better predict their activities. So I feel that we are really living in a time that everything is moving forward with almost exponential speed, and so I feel that it has to give sometime soon. I don't know whether it's going to be in the next 10 years or next 30 years, but I'm optimistic that we will find ways how to help these patients. And so the other question was whether this is just applied to ALS or whether this could be more generally applicable. And I think that, so as I mentioned before, protein misfolding is something which you find in most neurodegenerative diseases. It's actually different types of proteins that get misfolded and cause degeneration in, for example, Parkinson's disease than in ALS. But I think that if we go after the core pathways that are sensitizing the cells to misfolded proteins, like for example the MAP kinase pathway that we try to inhibit, that this might be relevant to other types of cells that are exposed to misfolded proteins. So our hope is that if we find treatment for one disease in this sort of category of diseases, we will be probably much better positioned to treat the other diseases as well.
Hello, and thank you very much. It's very fascinating. My question is, in your experiment, you injected a mouse at day 1, I believe, with the transcription factors. And did you try injecting a mouse at later points?
Yeah.
And was it successful?
Yeah, so this is another excellent question. So obviously when we inject mouse that is 1 day old, it's way before the mouse manifests any disease, and so that would not be really helpful for patients, because usually when patients get to clinic, already they have fully developed symptoms, and we estimate that probably up to half of their motor neurons are already dead. So we started now doing experiments where we are injecting the factors at later and later time points. And in our latest experiments, we actually found that we can deliver the factors at the time when the cell already has the misfolded protein aggregates, and expression of these factors is able to resolve them relatively quickly. So we think that it's not only protecting the cells or preventing the formation of the aggregates, but even at the time when the aggregates already form, that these factors are able to help the cell to clear them and start looking much healthier. So we have a hope that even coming late would be beneficial, but these experiments are still ongoing in the lab.
Thank you for your talk. One of the early slides you showed upper motor neurons and lower motor neurons in communicating to motor output. Is there a difference in the response to the misfolding protein response? And are both of them targetable by the possible therapies you showed?
Yeah, another excellent question. So all my talk was about the motor neurons that are found in the spinal cord, and that are the final step, the final output step to connect to the CNS to the muscle. But actually in ALS, you get also degeneration of the cortical upper motor neurons, and those cells are actually very different from the lower spinal motor neurons. So the spinal motor neurons are cholinergic, the upper motor neurons are glutamatergic. They use different neurotransmitters, and they rely on very different transcription factors in order to specify their identity. Yet in the disease, both of these accumulate misfolded proteins, and both of them will show degeneration. So I would say that in terms of the therapeutic approaches, the first one, the kinase inhibitor, my guess would be that if it worked in the lower motor neurons, it might help also the upper motor neurons, but we have not tested them. We don't have a good way how to make upper motor neurons from embryonic stem cells yet, and so we don't have good models for the disease. For the second part, where I talked about the reprogramming of the identity, I don't think that that would work for upper motor neurons, because, as I said, there's going to be different transcription factors that specify their identity, so we would have to tailor the cocktail of transcription factors and also the regulators to make sure that the factors are expressed in specifically these cells would be different than for spinal motor neurons.
Now, your animal models, I mean, you have identified, you said about 60 genes. You mentioned the SOD1 model. Does the prosentin work in that model? Does it prevent or retard the disease? That's question one. And the question number two, your clinical trial, I have a friend who now is suffering from the disease, and she's at your center. At which stage do you take patients?
So the clinical trial is, we have not yet started with ALS patients. We finished the 1A and 1B phase that was in healthy volunteers. The patients will start, and actually they will start in Europe first, because we got approval in Europe before FDA lifted the limits. So we'll be starting at Utrecht probably in June. And it's going to be, focusing on early stage patients as well. So unfortunately, I cannot promise that the trial will be, I'm hoping that we will open a site also in US, but not sure where. And the first question about the mouse model. So we tested prostatin also in the mouse model. And what we see is that when we treat the mouse at the time when there is already the first signs of pathology in motor neurons, and start treating with prostatin at that point, we significantly delay the onset of the disease, onset of the tremors and loss of muscle mass, and delay also loss of weight in these animals, suggesting that they have prolonged or that we delayed the onset of the disease. But unfortunately, we do not really prolong the total survival of these animals. And we don't know, because the mouse model is much more aggressive than human disease, because it kills mice within several weeks, while in human the disease goes on for several years. So I feel that it's important to test the drugs in patient population rather than try to extrapolate from the animal models. Yeah, there is not actually. We have 20 plus genes, but the other mouse models that people developed, none of them really recapitulates the pathology of ALS as well as this model. So it's another thing which is unfortunate, that we don't really have good models in the animals. You know, maybe it's because mice live such a short time that it's difficult to model correctly these late onset diseases.
What could be future of stem cell and cord application with artificial intelligence? Because we have from both our boys as a stem cell cord bank.
You know, I think that use of artificial intelligence is gonna be, or I would say more machine learning approaches is gonna be useful, because people are collecting more and more data. So you can now, as I said, you can derive these pluripotent stem cells from anybody, from skin cells or from blood cells. So people are building banks of stem cells that from people that have ALS. Actually, one thing that I really basically shoved under the carpet is the fact that majority of ALS cases are what we call sporadic, so they are not caused by mutation in a gene. So 90% of ALS patients will not have identified mutation. And so we don't know whether these patients have a combination of mutations that are just unfortunate enough to result in the disease, or there might be even some exposure to toxic substances in the environment. And so building large repositories of stem cells derived from these patients with both sporadic and familial diseases might eventually lead to some new insights. But I think good use of machine learning and AI depends on good quality of data, and I think that we still are struggling with basically collecting information about these cells that would be reproducible and robust from one lab to another, and so I think that those are kind of limitations.
Did you tell us what prosetin is?
Yeah, I did not really go into details, but it's inhibitor of this pathway. It's called MAP kinase pathway.
What is it, though? It's a chemical? It's a small molecule?
Yeah, it's a small molecule. I had the structure somewhere, but it basically blocks a cascade inside the cell. Yeah, here.
Oh, there it is. Right. I see. Okay.
It blocks a cascade that basically transmits information, you know, from the sensors that sense that there are protein misfolded in the cell, all the way to the nucleus that initiates then the degenerative process that kills the motor neuron. And so we think that if we interfere with this pathway, the cell will not undergo cell death or apoptosis.
And Prosetin is a village in the Czech Republic?
Yeah.
Is that where your people are from?
No, actually, I did not realize that there is a village called Prosetin in Czech
How did you
Republic.
pick the name?
It's complicated. It was actually called initially prostatin, which was built out of that is pro-survival factor that inhibits STE. Set of kinase.
Naming it after the village, your family would be better. It's a better story.
Yeah, I think that we have to change it.
Last question: do you believe that beta amyloid is the causative agent of Alzheimer's? You'll pass on that.
I don't know. I'm sort of subscribing more to the tau pathologies that are driving the, intrinsically are driving the degeneration inside the cell. I think that the tangles of the tau, phosphorylated tau protein is what the cell senses and kills it. I'm sure that the beta amyloid are contributing, but I would say that also the studies where there are effective ways how to resolve some of these amyloid aggregates might be.
Those are monoclonal antibodies. Lecanemab.
Yeah.
Are you aware that there's a blood test that just got approved to measure p-tau in the bloodstream as a possible early indicator of?
Yeah, so that's
a company called Neurocode.
Yeah, so that's another thing that people are like actively pursuing, finding basically biomarkers of these diseases that you could detect in plasma, because that, you know, again, in combination with a therapy, that would be great, but if you don't have a therapy, people don't really have drive to be tested for these diseases.
Well, that's why lecanemab, people are now thinking they can be tested and then treated.
Yeah. No, I think that's a good point. And so besides, so for ALS, is actually people use different biomarker called neurofilament light chain, which is shedded from degenerating neurons, and you can detect it also in bloodstream. So
right now, ALS is a clinical diagnosis.
Yes.
I apologize for the question. As you may expect, I was just wondering how does your work compare to your grandfather's work? You know, he also worked with kind of fibers, the nylon fibers. Kind of a dead material, but obviously he came to great results. And you are working with a living fiber or something like that. So I know my question is kind of silly, but how can you compare the two fields?
Well, that's a tough question. I think that I feel that I got, other things that's sort of my direct connection to chemistry from him. I feel that it's more maybe creativity and curiosity and sort of not getting discouraged by negative results. But also I cannot really compare myself to him. His impact on humankind was, is incomparable. He basically invented hydrophilic gels, which are gels that are biocompatible. So pretty much anything that we are now using as a biocompatible material comes from his research. And the soft contact lenses were just one of the clever ideas that he had. I wish I could do 1/10 what he has done, but I still have a few years to go.
I wanted to ask about Guillain-Barré syndrome, which is kind of a recoverable motor neuron problem that I wonder how that's related, or if it's related, and whether it would be of interest to you to find out how those cells recover.
Oh, I have to ask my daughter Clara, what is Guillain-Barré syndrome? Can you? Is it interferon? Interferon? Yeah, I feel that it's challenging because even despite everything that I read about ALS, I still cannot tell you really what is the pathology that goes from the misfolded SOD to the cell death. What are the processes inside the cell? And I think that's true for absolute majority of these diseases, that we don't really know what happens. And it's unfortunate because there is not really easy way how to study these pathologies. There is, we don't have access to the cells, so you rely on either stem cell derived neurons as we do, or on trying to build animal models, but the animal models often are imperfect, like in ALS.
You mentioned that ALS is sporadic. Do we have any statistics on the geographical prevalence of ALS?
Yeah, there were a number of studies done,
Speaking
public
thinking
health,
of environmental factors.
Yeah, epidemiological studies where people tried to identify sort of sources of potential toxins. There are, different hypotheses. So there was a cluster of people that were affected by a disease that looked like ALS on the island of Guam, where they identified some seeds that have toxins that could potentially potentiate the disease. There was a cluster of increased ALS in veterans that came back from the first Gulf War, and there was thought that they were exposed to maybe some neurotoxins, organophosphates. There was a cluster of Italian soccer players, for what we know, and again there was hypothesis that maybe pesticides used in the grass could contribute to the disease, but it's I think the epidemiological signatures are not strong enough to really make a very strong and conclusive argument for these. Agricultural workers are also sometimes argued to have higher incidence of ALS.
I'm 20 years out of the field, but it's a big family, right? And do you know which particular ones you inhibit, and do you know which are the target genes of those particular kinases?
Yeah, so so you are correct that MAP4 kinases, there is in excess of 20 different MAP4 kinases, and we profiled actually all kinases in the, or not all, but I would say around 400 different kinases in the genome, and tested the drug and mapped which are inhibited. So out of the MAP4 kinase family, there are 5 MAP4 kinases that are very strongly inhibited. And besides MAP4 kinases, there is, I think, 3 other protein kinase D, which is inhibited, and CSF1 receptor, which is inhibited, that potentially might also play a role in the disease.
And you know the targets of those 4 kinase inhibitors?
Oh, what's downstream? Yeah. Yeah, so in the end, we believe that this pathway then converges on a transcription factor called c-Jun. And when it becomes phosphorylated, it translocates into nucleus, where it activates target genes that will then result in the apoptotic process in the cell death. But again, as with everything, there is these pathways have many different branches, and it's fairly complicated. So I would not say that this is the only explanation how this drug works.
I have my one question as a literature scholar. Was that really lovely 2-part alchemy metaphor your own invention, or did you come across that somewhere else?
No, I think it came when I was basically brainstorming the title for this talk with Susanna Halsey, and we came to alchemy somehow, so I had to adapt my presentation to include that.
I think it works really well. You should stick with it.
What you delivered to us is very interesting. Thank you for this. But I would like to ask you a question, because looking at the whole presentation, I noticed a couple of holes, what I don't understand. I would like you to explain.
Sure.
You're calling the stem cells pluripotent, right? Do you know if the stem cells have programs, each one of them?
So the stem cell itself, every cell has a program, and that's what defines the cell function and the cell properties, the cell biochemistry. So even stem cells have their own program, and actually you are using the transcription factors that define the stem cell program to reprogram the skin cells into stem cells. So the new technologies that I mentioned, that you can take samples from any one of us, actually I took a little biopsy of my skin and actually converted these cells into stem cells. I use the transcription factors that activate the stem cell program in these.
Yeah, I understand. But inside of your body, you've got so many different types of stem cells. Do you think they have the same program?
Oh, no.
Everyone have them different?
Each type of stem cell will have different program inside the body.
Okay. Can you change that program?
Well, you can use, as I showed, you can build these viruses that if they infect the cell that you want to change, you can deliver into the cell different transcription factors that will change the cell.
Playing with viruses is kind of dangerous stuff, right?
Yeah, there is very few viruses that are approved therapeutically at this point, and most of them are for very serious diseases. So.
Okay. Do you know what the very small embryo like stem cell is? What kind of cell is that?
Yes.
You do?
Yeah.
Okay. So very small embryo like stem cell is the stem cell. We not call it the stem cell because it's embryonic, has no program. The question is, in your process of trying to heal that kind of disease, can you use those embryonic stem cells? Not stem cells yet, because they have no program. You can give them the program, and they automatically be the one what you need.
Yeah, so your question, you're asking whether we could use the stem cell
not stem cells, very
no, the
embryonic
motor neurons derived.
like stem cell,
Yeah.
because they have no program, so we cannot call them stem cells. Yeah. But this is the cell without program. If you give them the program, they become a stem cells for the things you need.
I think these are interesting questions. We should probably discuss this after, because it will be for long discussion.
Okay.
A different topic. Could you tell us about the funding sources you get for this type of research? And to what extent are pharmaceutical companies involved? And are you getting the funding that you need?
Yeah, these are great questions. So we have, I listed some of the funding. So for a lot of the work that I have done is, I think the main funding that we get is from two sources. One is NIH, and then the second source is actually a local foundation called Project ALS, that is a New York City foundation that funded my research for now, in excess of 20 years. And they really, I think, made me more interested in working in translational biology. I was always much more considering myself a developmental biologist, interested in how cells work, how development works, trying to understand how transcription factors control cell identity. But with Project ALS funding, they sort of allowed me to build a part of my lab that is working on these more translational or applied clinical topics. And so I think that helps a lot. Obviously, there is never enough money. So even with Project ALS, they helped us to bring the small molecule all the way to get approval from FDA, the IND, to start the clinical trials, but they were not able to support us in the clinical trial. So that's why we had to find investors and start a biotech company. That was the only reason I'm not interested in running biotech, but that was the only way how to basically raise funds to go into clinical trials. So yeah, it's challenging, constantly applying for grants, and now actually my last few grants got just not funded by a single percent point because there was continuous resolution, and I had to reduce the budget, so I have to go into another cycle, which delays all the funding by another 6 months. So it's, unfortunately, that takes me away from doing interesting science and just doing the bureaucracy by the computer.
Since we are arts and sciences organization, are you aware that you're creating with those images like really works of art? There was a slide with those 3 pictures. Can you go back to that maybe? It was like 3 paintings. Yeah.
This one?
Yeah. Are you considering doing an exhibition or selling those? Or
yeah, maybe that's how I could raise money for my research. Yeah, no, it would be nice one day maybe when I'm old enough. And well, thank you very much, everyone.
All right, thank you so much, Hynek.